Moving into metaphase, the chromosomes will start to condense. Condense means they get shorter and they coil even more, and then they align in the middle of the cell. Look at this picture right here. You would see by this time centrioles will have moved to opposite poles. You've got the north and the south pole, if I may call them. As they moved to the opposite poles, spindle fibers are formed, and quite miraculously, spindle fibers attached to the centromeres of each chromosomes like this, causing these chromosomes to line up at the equator of the cell. We call this the equator, meaning the middle of the cell. So this marks metaphase. So if they ever ask you what are the points to include in metaphase, you can say that each centriole reaches to the opposite poles, spindle fibers are attached to the respective centromeres of the chromosome, and most importantly, chromosomes line up across the equator of the cell. Okay, so you've got the centromere right here. As we head on to anaphase, you would see later that the sister chromatids, you remember what sister chromatids are? They are genetically identical strands of DNA on a chromosome. So in anaphase, this sister chromatids will say goodbye to each other. It will separate. The chromatids will be pulled apart like this because the spindle fibers which are attached to them will pull these chromatids apart. Recapping, see the spindle fibers here? As they shorten, they pull the sister chromatids apart. Centromere will be heading towards the pole. So if you were asked to draw the chromosome, how will it appear under microscope during anaphase? Make sure you draw the centromere as leading to opposite poles and the chromatids at the back, pretty much like this. What to write down during anaphase? Chromatids move to opposite poles, centromeres first, pulled by the microtubules. Microtubules is the same as your spindle fibers. Now in telophase, nucleus and nucleolus start to reform. Sister chromatids unravel. Unravel means they start to unwind and uncoil to form chromatin. Do you remember how chromatin are actually just strands of DNA around histone proteins? This will be followed by cytokinesis. Okay, so telophase and cytokinesis tend to overlap. You don't have telophase first, nicely followed by cytokinesis, but rather they happen almost at the same time. Telophase is distinguished by the start of the formation of the nuclear envelope or the nuclear membrane and even the nucleus like this. While cytokinesis will show that the cell's cytoplasm start to constrict or start to form cleavage, hence dividing the entire cell into two. Eventually what's going to happen is you'll have two cells with genetically identical DNA. So there you go. We're done with all the phases, the PMAT phases, prophase, metaphase, anaphase, and telophase, followed by cytokinesis. In your AS syllabus, you only need to know about mitosis. You will only learn about meiosis in your A2. This is another picture that shows you cytokinesis. It's an electron micrograph of a cell. This is cell number 1. Look at the nucleus here, and this is cell number 2. How do you know it's cytokinesis? The nucleus are already properly formed. You don't see strands of chromatids anymore. Number 1. And number 2, you see there's a cleavage between the two cells' cytoplasm. These are other pictures of the different phases during mitosis. So we're going to begin with interphase or the beginning of mitosis. And this is prophase. Notice the difference between this picture and this. The nuclear membrane disintegrated here. So that's how you know this is prophase. And then over here is metaphase. The black line that you see here is actually the chromosomes aligning at the equator of the cell. And then this is anaphase, where sister chromatids are separated and then they move to opposite poles. And this one over here is telophase. How do you know it's telophase? You don't see the nucleus like in cytokinesis. You start seeing the nuclear envelope, right? This is chromosomes. They haven't formed nuclear envelope yet. And that's how you know the difference between telophase and cytokinesis, by looking at the structure or the absence and presence of the nuclear envelope. Telophase doesn't have the nuclear envelope yet, just chromosomes aggregating at the center like this, while cytokinesis have a proper nuclear envelope forming around the uncoiled DNA, which is now known as chromatin. So before we end, I thought we should make this clear, the difference between centromeres, centrosome, and centrioles. So centromere is really what connects the two sister chromatids together. And you have this term called kinetochores. Kinetochore is actually a point where the spindle fibers are attached to the centromeres of the chromosome. On the other hand, centrioles, centrioles, plural for centriole. When you put them together, when you have two centrioles together, we call them a centrosome, and they are responsible in the formation of microtubules, or also what you know as spindle fibers. So that's the end of mitosis as a whole. The next lesson, we're going to look at the significance of mitosis. Why should we bother about mitosis?